Let me just echo my thanks and my real pleasure to have this chance to come down here and participate in this wonderful meeting. I want to step back a little bit. We talk about tumor heterogeneity, but here's the big picture. Tumor heterogeneity is in the context of the fact that these neoplasms are evolving in our bodies by somatic evolution. So this is the kind of cartoon I think about that comes out of the evolutionary biology literature, that there are clones that are mutating and that are growing and shrinking either randomly by genetic drift or by natural selection. And in fact, this means that all of the, or most of the whole theory of decades worth of research and thought in evolutionary biology can be applied to cancer. So that means natural selection, mutations, genetic drift, cell migration, and that the rate of this evolution is controlled by the parameters of that evolution. That is the mutation rate, the population size, the generation time, and the clonal expansion rate, or if you're an evolutionary biologist, you can think of this as the fitness differentials between the different clones. So that gives us a lot of machinery that we can apply to cancer. And here's some of the estimates of some of the parameters involved. So mutation rates, of course, there's all different kinds of genetic alterations in tumors, and none of these numbers are very well set yet, but it's good to do a little bit of sort of back-of-the-envelope calculations here to get at some intuition about the evolvability of these neoplasms. So point mutations might range from 10 to the negative 11 to 10 to the negative 7. Chromosomal abnormalities seem to be easier to happen. The generation time, we don't have great data on, but maybe 1 to 3 days for the stem cells, cancer stem cells. And that we have evidence that some of these neoplasms take many decades from their first initiating hit to the point that the patient feels sick. And so that leads to either between hundreds to tens of thousands of cell generations in the history of a tumor. And population sizes is better known, on the order of billions to trillions of cells in a tumor. And you put these things together, and that means that pretty much every possible base pair in the human genome is mutated somewhere in a tumor every cell generation. Right, so every possible mutation is being explored pretty much every day in one of these large tumors. And then it's not a surprise that it can discover resistance mutations, and that famous picture of the melanoma patient on the BRAF inhibitor had resistance mutations in each of those large lesions across his chest. And then another important point to get here is all this evolutionary processes happening in the complex ecology of the tissues. So that means there's important interactions with the stroma, and we see co-option of different stromal cells, the fibroblasts, the macrophages, et cetera. And we also see, of course, immune predation. We see interactions both positive and negative for the tumor with the immune system. And then there's also interactions, of course, between the clones. And Nellie showed some really fascinating example of cooperation between an angiogenic clone and the sort of free-riding other cells in these tumors that she was creating. Clearly, there's competition between these clones and maybe parasitism, that free-riding. And as I mentioned yesterday, we don't have a good grip on what the limiting resources are in these ecosystems. It probably changes over time, pre and post angiogenesis, and also in different areas of the tumor. So there's a whole complexity of ecology, which is essentially what microenvironment people are doing, but we really haven't applied both ecological theory or the systematic approaches to understanding that ecology. So I think of every tumor is obviously unique. It's a snowflake. It's on steroids. It starts from the unique genome of its host. It accumulates hundreds of thousands of alterations. So you get this mosaic of clones, and the mosaic of clone is overlaid upon a patchwork of microenvironments that are probably selecting for different clones in the different microenvironments, whether it's hypoxic or near a blood vessel or in a bone niche or whatever. And so this means that one thing that's come up a number of times here is that these tumors don't have a genome. It doesn't really make sense to say I sequence the genome of a tumor. Right? A tumor has billions of genomes. They're related to each other, and what you get from the sequence, of course, is the majority clone or the frequent alleles at any location, but not a single genome. So this is the sort of view of what's happening in tumors that are evolving by natural selection with this complex ecology. This is sort of what we do in most of cancer biology. We have a single time point. We take a single sample from this tumor. We grind it up, and we do some assay on it. And what we're talking about today and yesterday is this. We're looking at the heterogeneity in a tumor at a single time point, right? And it's very illuminating, right? You can't understand what's happening without understanding the interactions and the complexity of the different clones that are in there. But it's still only a small part of the picture. That is, we're not seeing how it's changing over time. So in fact, I would say one of the biggest open areas in cancer biology is we don't understand the dynamics of this process. We don't understand, in general, the rate of the clonal expansions. We don't understand how often new viable clones are appearing. We don't understand the diversity of these microenvironments. And we don't understand the process that maintains this intratumor heterogeneity. So I'm using ITH as an abbreviation for intratumor heterogeneity. So as I think it was Nelly pointed out yesterday, that when you have a spatially structured population, it often inhibits clonal expansions or expansions of new alleles. If you have specialization of clones, you can maintain diversity. We don't understand the cooperation or the parasitism that might be doing this. And I put up this figure from, let's see if I can get the pointer up here. No. Yeah, I'm using the mouse. It's actually nothing's appearing. Okay. The figure in the lower right-hand corner is from Bob Gatenby's work, where you can actually use multimodality MRI data to try to non-invasively characterize the different kind of habitats that are within a tumor, that is, the different microenvironments, which may be a non-invasive way of starting to characterize some of this heterogeneity, or at least the environments that are putting different selective pressures on the cells. So what's the importance of intratumor heterogeneity? Well, one of the major important parts of it is that it's the fuel for natural selection. You can't get populations of change if you don't have variation within that population. And of course, we get this from these somatic genetic lesions and also epigenetic lesions that were mentioned yesterday. That intratumor heterogeneity is also important in that it's the information that's being passed from generation to generation. You have to have heritability to get natural selection working. And then, of course, the engine of natural selection is this fitness differential, is how that heterogeneity affects reproduction and survival of the clones. So what are the limitations of the current data? Well, I'm sure you're well aware of this. As I mentioned, we take one sample typically from a tumor. That's a mix of clones that are difficult to deconvolute. So Noemi Ender, who's sitting here and has a poster, has one of the best algorithms for trying to deconvolute those subpopulations or subclones called expands. We also heard David Hausler talk about his algorithm for doing it. It's similar yesterday. When we look at the diversity within tumors, what we typically see is this kind of exponential distribution. This is some data from lung adenocarcinomas. And the divergence measure is one of the measures of diversity. You can see that most of the tumors we looked at had very low divergence. You see that big spike on the left-hand side. Then you have this long tail of some tumors that are highly heterogeneous. And then if you look inside any one tumor and try to characterize that diversity, there's probably a very long tail of rare clones. You saw that mention of the paper from Navin, that's the Wang paper there, yesterday, where it looks like every cell is different. So if you really got down to the fine-grained detail, you would see a very long tail of rare clones and then some larger clones that have dominated or are more easy for us to detect with current methods. So what's the importance of intratumor heterogeneity? Well, there's sort of, I think of it both as a scientific importance and a clinical importance. So the scientific importance basically falls to phylogenetics, and the clinical importance basically falls to resistance, and I'll talk about each of these things. So one of the main innovations that's been led by Charlie Swanton is taking multiple samples from a tumor. And once you do that, you get huge power to bring in all this machinery from evolutionary biology based around phylogenetics. So if you build a phylogeny, this is one of the phylogenies from our patients, one of our Barrett's esophagus patients, where we've had 12 biopsies from this patient. Once you can build a relationship between those cells that you've sampled from the tumor, that gives you the history of the neoplasm, which allows you to figure out the order of events along the different branches. And you should note here that a tumor doesn't have a single order of events. There's an order of event along each of these branches. So it's incorrect to characterize a sequence of events in a tumor unless you happen to be lucky enough that there was a clonal expansion that went to fixation, took over the tumor, then another one that went to fixation, took over the tumor. And you would see that in a phylogeny if it actually happened. But so far the evidence is that that does not in general happen. Doing the phylogeny gives you the clonal structure of the tumor. It allows you to see clonal expansions. You can see this branch in the bottom part with biopsies 13, 8, 10, and 11. Those were all a clone that had had 19% of the genome messed up by copy number alterations. You can detect convergent evolution because the phylogeny will show you that the same mutation happened on independent branches. You can actually measure the mutation rate, and I'll talk a little bit more about that later from a phylogeny if you have at least two time points. You can measure the population size, so in this case the number of cancer stem cells. You can estimate the time since initiation if you have a rough molecular clock that you're building your phylogeny from. You can then time the divergence of particular clones if you're interested in it. and also look at the nature of therapeutic resistance. And by this, I mean that if therapeutic resistance is being driven by selection for a particular mutation, you'll see that branch of the phylogeny expand. If resistance is based on the fact that you just are not killing cancer stem cells, then you won't see any effect on the structure of the phylogeny after therapy. The same diversity will persist after therapy, and you can distinguish those two different hypotheses for the nature of therapeutic resistance. I want to mention that the evidence, I sort of hinted at this, the evidence so far is that you don't get full clonal expansions that go to fixation. That is, they don't take up the entire tumor, which means that ancestral clones linger. And that's great scientifically, because that means if you sample those ancestral clones, you can actually reconstruct the whole history of the tumor. This is some evidence from a variety of papers. In the lower left is one of Mel Greaves' papers. As showing what I've circled is the original the sort of ancestral clone. The upper figure is from Simon Leadham in Barrett's esophagus, showing that some of the crypts had no mutations at the locations that they're looking at. And you've seen the figure on the lower right from Mulligan in ALL, showing that often resistance derived from ancestral clones. I also want to get to the clinical side of this. I want to point out that every drug that's ever been invented in cancer selects for resistance. Right? This is not a problem that we have ever been able to avoid. And it's quite possible that every drug that ever will be invented for cancer will select for resistance. And that's an open question, of course. But this is the big clinical problem for cancer. So I should point out that it's not enough to just target the dominant clone in a tumor, which is sort of the sort of foundation medicine approach of just let's see what the dominant clone is and find a drug for it. Of course, that will select for the minor clones that don't have your drug target. It's not even enough to target a trunk mutation, that is, a mutation that's in every cell of the tumor. And of course, the canonical example of this is Gleevec targeting the BCR-ABL lesion. It's the foundation trunk mutation in CML. And there have been over 100 different point mutations or alterations in CML that provide resistance to Gleevec. So he doesn't get away from the problem of resistance to be able to target all the cells in the all the cells in the neoplasm. And the figure in the upper right-hand corner is just some data from Neil Shaw in particular patients showing the point mutations that were caused resistance to Gleevec in CML. So these resistance mutations abound, and the evidence so far is that in general they're present prior to therapy. And so what you're doing is selecting out those clones when you apply therapy. And it's like the phoenix from the flames, the tumor comes back. Now, we've been talking a lot about genetic diversity, and I want to make a point that of course selection happens on the phenotype of the cells, so it's actually phenotypic diversity that's probably even more important than genetic diversity, although it's the genetics that propagate that resistance to the next generation of cells. And this phenotypic diversity brings in the issue of the plasticity of the cells, that the same clone may be able to take different phenotypes, and particularly if it has a sort of a stem cell-like flexibility, it may be able to take a large variety of different phenotypes. And we don't fully understand that yet. So a lot of the biomarkers in cancer have been based on measuring the products of this stochastic evolutionary process. And there's a lot of problems with that. So we can make panels of various mutations or proteins that may come up. But so far the evidence is that these tumors, of course, are spatially heterogeneous. So where you take your sample may not hit the relevant clone that determines the future of that tumor. Many of the mutations are, of course, they're stochastic, so they're historical accidents. The evidence so far is that there are many different ways of building a tumor, many different sets of mutations. So if you have a particular mutation, you may only be predicting the relevance of a minority of cancers of that type. Of course, since it's stochastic, the critical mutation that determines the future of that tumor may not have happened yet when you take the biopsy. So these are all major limitations in using the products of evolution as your biomarkers. So instead, we argue that there's a set of universal biomarkers based around measuring the process of evolution, the evolutionary process itself. And that's based on measuring those rates of the parameters of the rates of evolution. So if we could measure the mutation rate of a tumor, the stem cell population size, the stem cell generation times, and the clonal expansion rates, these measures should be applicable to any type of cancer and should relate to the chance that cancer or that neoplasm is going to either progress to malignancy, or if it's already malignant, the chance that it's going to evolve around your perturbations, that is your therapy. It's also the case that the ecology of the tumor structures this evolution, and so there should be some universal biomarkers based on ecological theory. That is, what are the selective pressures? If you take for a moment the sort of Jurassic Park view that life will find a way, then you can ask the question, what are the selective pressures? And you can bet that the tumor will respond to those selective pressures and predict its future behavior based on just understanding the pressures. So again, limiting resources, the sources of cell mortality. I bring up this because it's the basis of life history theory in ecology, that different sources of cell mortality or different and pressures on the tumors will select for either fast-growing, sloppy, mutating populations or slow-growing, highly competitive populations. And of course, environmental heterogeneity is part of this as well. And I'll show you some work later that predicts that the microenvironmental heterogeneity should select or not for metastasis. The figure on the right, I'm going to go into a little bit later, but some work of ours actually measuring mutation rate in Barrett's esophagus and how it's changed when people change their NSAID use. So in fact, here's the theory behind this. So in evolutionary biology, typically we only have one time point. We go out into the field, we sample a bunch of individuals of the population, and you can describe the genetic diversity of that population. Unfortunately, the data can be explained, the data is usually characterized by this parameter theta, which is 4 times the mutation rate times the population size. And the intuition here is that if you build a phylogeny of the relationship between the individuals you sampled, you can explain the genetic diversity by a small population with a high mutation rate or a large population with a low mutation rate. It will give you exactly the same data. So you can imagine, you can see the sort of dotted line there. You can imagine stretching this phylogeny back and forth depending on the size of the population and the mutation rate. And as those scale together, you can explain the same data. Now, if you take 2 time points, you fix the scale of the branches on that phylogeny, and you can no longer stretch it. And mathematically, that just means you can deconvolute the mutation rate from the population size. So it was this work by Alexei Droman that inspired us to apply this to cancer. They had done it in evolving viral populations. I guess I can just go back to this figure and say that what we found when we did this in Barrett's esophagus, we had 2 time points when the patients were on NSAIDs and 2 time points when they were off NSAIDs, and we were able to estimate their mutation rates on those different regimes and show that in most of these patients that when they went on NSAIDs, their mutation rate dropped by an order of magnitude. And actually, the mutation rate here is copy number changes, so it's the acquisition of amplifications, deletions, and loss of heterozygosity, not point mutations. All right. So one of the major universal biomarkers we're interested in is measuring and quantifying the amount of intertumor heterogeneity, which is essentially, I think, why I was invited to this meeting. And our hypothesis here is that the more diverse tumors, these are premalignant tumors, are more likely to acquire the remaining hits to progress to invasion. Right, so the more diverse tumors are basically more evolvable is the hypothesis. And so I've mentioned Barrett's esophagus, but here's the background on it. This is a great system for studying these processes. So Barrett's esophagus is associated with chronic reflux from your stomach. Most patients have a hiatal hernia, so they don't have a very good valve at the top of their stomach, and they get their lower esophagus bathed in stomach acid every night. And in some of the patients that have chronic reflux, their tissue will actually change. You can see this reddish area through the endoscope. And instead of the multi-layered flat squamous cells of the esophagus, it changes to look like the intestine, where it's a single layer of cells, and they have a structure like a crypt, these sort of well-like structures. And it's only one layer of cells, so you can see the blood vessels behind them, and it has this reddish appearance. It's interesting for cancer because this condition of Barrett's esophagus predisposes for esophageal adenocarcinoma with a relative risk of somewhere between 30 to 120 compared to people who don't have Barrett's esophagus. So that looks scary, but it's not quite so scary because they've actually looked at how often people progress to the cancer. It's less than 1% per year. So most of the people with Barrett's esophagus will never progress to esophageal adenocarcinoma and will die of some other cause eventually. Now, the only definitive cure for Barrett's esophagus itself is esophagectomy, that is cutting off the lower part of the esophagus, hitching up the stomach, and sewing it back to the remaining esophagus, which is a very difficult surgical procedure. And if you go to the high-volume hospitals with real expert surgeons, like at Stanford, they'll kill 8% of their patients from that procedure. Right. Most of these procedures are not done in high-volume hospitals, and there's more like 23% mortality at the low-volume hospitals. So you put those numbers together, and it's just not worth the risk of removing Barrett's esophagus because you're not generally going to die of this cancer. So instead, what they do is serial endoscopies. So they put the endoscope down, and they take multiple biopsies looking for cancer, and then they send the patients home and ask them to come back a year or two later, and we'll look again. So that's the typical clinical management of the disease, which means that we have this great scientific opportunity because not only do we get multiple samples across the surface of the tumor, but we get multiple time points. So we can watch over both space and time what's happening here. And this is more like many indolent neoplasms, DCIS in breast, many low-grade prostate tumors. The question is, which of these patients are going to progress to cancer? That is, we need to stratify the high-risk patients from the low-risk patients and focus our resources on the high-risk patients and reassure the low-risk patients, don't worry, come back in 5 years instead of 1 year. So one of our inspirations here then is to measure this process of evolution, to do this risk stratification based on these universal biomarkers. So here's the little diagram there that we're getting multiple time points, multiple samples across space, and we can watch this evolutionary process. So we used these measurements from ecology to quantify species diversity in ecosystems, and we applied it to measuring the clonal diversity in these tumors. One of those measurements is genetic divergence. So this is just if you take any two samples from the population, how genetically different are they? Or in this case, what proportion of the genome differs based on the SNP array assay. And what we found in this cohort of over 200 Barrett's patients was that the people that had the highest divergence, highest diversity at baseline were more likely to progress to cancer than the low divergence. And it turns out that it doesn't really matter how you measure the genetics of the clones. So you can define a clone based on measuring driver mutations like P53 or P16. And that's the Kaplan-Meier curve on the left. Or you can measure the passenger mutations like expansions and microsatellites in non-coding regions that look like they should have no phenotypic effect. But those neutral mutations also mark the history of the clones and can give you estimates of the underlying genetic diversity that's relevant to the tumor. And indeed, you still get statistically significant predictions of progression from measuring the neutral mutations. So it doesn't look like it matters how you define a how you define a clone. It also looks like it doesn't matter which of the hundreds of different measurements or metrics of diversity you use from ecology. You can just count the number of clones, that's the lower left. You can look at divergence, you can look at Simpson's index, you can look at Shannon's index. It turns out it doesn't matter.
Carlo, you're showing the cancer incidence rate but not when it occurs.
Yeah, no, this is when each step in these Kaplan-Meier curves are when we detect the cancer in that patient. Yeah. Yeah, you're probably more used to looking at survival curves. So these are exactly the same. They're just flipped upside down, incidence curves. All right, so that was in the Seattle Barrett's cohort. And of course, we don't change clinical practice until we see it replicated in an independent cohort. And I'm happy to report, although this hasn't been published yet, that we've used a cohort of patients from Mayo Clinic that had high-grade dysplasia, totally different assay, a FISH assay, so we're looking at copy number changes at a few loci in single cells. But in this cohort, we have 3 different panels of FISH probes. It's called PS1, PS2, and PS3, so a total of 12 different loci have been looked at, but only 4 at a time. And you can it's semi-automated quantification of the number of copies of these different loci based on these different fluorescent markers. And then you get data like this, and you've actually seen some of this yesterday, that for every cell, there's every row in the spreadsheet, and you get the copy number of the loci. And so you can define a clone as the pattern of copy numbers across these 4 loci. And then you can count the frequency of each clone by the number of cells that have exactly the same pattern, and you can apply the ecological measures based on this. And so if you do Shannon's diversity index based on these probe sets, you get a statistically significant prediction of those patients that are going to go on to cancer. This Sheila Krishnadath is the PI of this cohort that was measured at Mayo. She's now at Amsterdam and has also a large cohort of earlier stage patients there that we're working with. Trevor Graham and Vitska are the people that have actually done the analysis here. So that was Shannon index of diversity. You can use another index like the divergence index. Again, predicts progression to esophageal adenocarcinoma in a totally independent cohort, totally independent assay, in fact. This cohort actually is high-grade dysplasia patients, so they're actually getting interventions. And despite those interventions, we're still seeing predictive power for progression. All right, any questions about that before I move on to the next one? So actually, I do prefer the questions during the talk than after. So I appreciate Daniel's.
Is this, it's hard to follow the slide, but is this validation the same degree of predictive power as your first?
Oh, no, we haven't quantified the degree of predictive power here.
Can you repeat that?
Sorry, so the question was, is when we do this validation of in this Mayo cohort, is it the same degree of predictive power as in the Seattle cohort? So we haven't actually tried to quantify the degree of predictive power. I'm just saying that there's little p-values you have in the upper right-hand corner there that it's statistically significant, but how close it is, it's hard to say. It's a totally different assay, so if you're asking sort of the relative risk or something, we haven't actually looked at that yet. Yeah.
So did treatment have no effect or equally decreased the risk in all risk groups?
So the question is, did the treatment have no effect or did it equally decrease the risk in all the groups? The answer again is actually we don't know. So we actually have totally ignored the treatment here and we've just looked at this measurement and outcome. So this hasn't been published yet, so maybe we'll have a chance to dig into that. But it's hard to do because these patients are getting multiple treatments and
You could just say any treatment, because if it's not affecting it, then
right. Yeah. So certainly the treatment is not based on whether or not how diverse they are, because that measurement was not available to the clinicians. Yeah, Daniel.
Going back to what you said earlier, so the various ways in which you could be diverging more. One is more divided
Yes.
generations, and the other one is higher mutation rates. Do you have a sense of which the relative goals of those?
Yeah, so Daniel's question is, divergence can be generated by just faster division, so you get a higher mutation rate per unit time because you're dividing more, and every division you have some mutations, or it could be generated by a higher mutation rate. No, we don't have a sense of which of those two is driving this yet. Good question. All right. So that's all in taking a pre-malignant neoplasm and predicting which ones are going to go on to invasion. We also believe that the amount of diversity in a full-blown invasive cancer should be a good predictor of survival because the more diverse cancers should be the ones that are more likely to harbor resistant cells. And that's true even if you resect the tumor. We predict that the more diverse cancers are the ones more likely to leave behind resistant cells. So at UCSF in the thoracic oncology lab, we have a large tissue bank of lung cancers, and we were able to take our tissue bank and start looking at diversity predictions for overall survival, not of progression to cancer. Let's see. So again, this is work by Trevor Graham, also Natalie Louie, who is a really talented fellow and surgeon in the lab. And we did the cheapest thing you could do. So to measure diversity, you need at least 2 samples to see how different they are. So we took 2 samples from each tumor, and we have the normal as well, so we can look to see what areas of the genome have lost heterozygosity. We apply SNP arrays to each of these samples as well as the normal tissue, and just calculate the divergence between the 2 samples, which again is just the percent of the genome that differs based on loss of heterozygosity. It's not a huge cohort. It's about 60 patients, 58. Most of these are early stage. You can see on the right they're either stage 1 or some stage 2, and that's just from the fact that you don't tend to resect stage 3 and stage 4 cancers. There's a pretty even distribution across grades. And of course, the patients are relatively late in life, an average of 70 years. So we also only took tumors that we had at least 5 years of follow-up, so we could get good survival data on them. Now, I want to make a point here, and this was alluded to yesterday, that there should be some correlation between the total mutational burden of a tumor and the amount of diversity in that tumor. It's not exactly the same. So the correlation in this data set is 0.58. And here's examples of, on the left is so there's a Circos plot for each of the 2 samples from the tumor, so in green and in blue. And what you can see is that a large proportion of this genome has been altered, but both sides of the tumor where we took the samples are basically identical. So high mutational burden, low divergence. Right? So that's the discordant case from your intuition. On the right is the kind of thing you would expect, that is a lot of lesions, but a lot of differences between the 2 samples. So high mutational burden, high divergence. Okay. So and here's the survival curve for these patients. So the most divergent tumors, the ones with the most diversity, are in the green line. That's the upper quartile of diversity, and the blue line is the patients with the lower diversity. So so what we're seeing is an overall survival dramatic difference between these 2 groups.
This
Yeah, that's right.
Focused at the cancer gene panel, or is this?
No, it's whole genome. It's a 2.5 million SNP array. So.
What are you really measuring there? The chance that one of those places getting hit seems low to me.
So here's, I mean, you can see a lot of the genome is being, is
You're measuring
being.
LOH, basically.
Yeah, that's right. We're doing
You're measuring LOH.
LOH. That's right. Yeah, loss of heterozygosity or deletions and amplifications. On the right is the multivariate Cox model of proportional hazards, and on what you're seeing at the top is that genetic divergence was the most significant predictor of survival in this group. Of course, age is a predictor of survival as well because we die when we're older. And stage is also significant, but not nearly as significant as genetic divergence. And so that looks great clinically, but of course the big caveat here is we've got a massively skewed cohort to early stage, so we don't have a lot of statistical power to see the relevance of stage in this group. Of course, stage 4 cancers are much more deadly than stage 1. Now, we also wanted to do something that David was talking about yesterday, which is even within a single sample, there's some signal of the amount of diversity within that sample. And this is the sort of cartoon of it that Trevor made, which is on in sort of panel A here. If you take a sample from a tumor and you hit 2 clones, then the lesions you'll see in that assay will tend to fall into 2 clusters of the frequency of an allele, a mutant allele, right? Because those 2 clusters are the mutations that are in those 2 different clones. On the right would be a very diverse tumor. You sample a lot of clones, and then you should see a lot of variance in the allele frequencies of the lesions. So we just did the stupidest thing you could do, which is measure the variance in the allele frequencies of the lesions we saw in the tumors, and asked if that was a predictor. And the answer is yes, although there's a little caveat here. We've got 2 samples, so we've got a within sample diversity for 2 different samples from the patients. And if we took the maximum of those 2 samples, that's what that data is there. So the maximum within sample diversity was predictive of overall survival as well. And if you actually ask for the average of the 2 within sample diversities, or just use one of them, you don't get nearly as good statistical power. So there's still a lot of value in taking more than 1 sample. All right. Now, this is not our data, but corroborating data from head and neck cancers that they use a math score, which is actually a nicer and a more sophisticated measure of within sample diversity using exome sequencing, and showed that the more diverse exomes, again, single samples, were predictive of overall survival in head and neck cancer.
Can I ask something about that?
Yeah.
Were you using LOH as your metric in the lung samples?
Mm-hmm.
What you're measuring is the rate of large-scale genomic changes, structural
Yeah,
variation
that's right.
in the tumor. And that's quite different than the mutation rate.
Right.
Say something about.
Yeah, so let me repeat that for the recording. So Steve is pointing out that what we're measuring with the SNP arrays are these chromosomal changes, loss of heterozygosity and amplifications and deletions that happen over large sections of the genome. And those have very different effects than a point mutation in the genome. And so can I say something about the relevance of that? So I would as you probably know, there's a big debate about whether or not there's an increased point mutation rate in cancers. So there's this big fight between Larry Loeb and Bert Vogelstein, et cetera, about point mutation rates. There is not a fight about chromosomal alterations. Cancers clearly have massively increased chromosomal alterations. So I think the evidence across cancers is that chromosomal level alterations are important and big and have massive perturbations to the genome.
Do you know if the rates of them are higher, or just the chances of them surviving and going on to survive further?
I don't have a quantification of whether the rates of those chromosomal lesions are higher or not. I can't imagine that. Yeah, I can't imagine that a normal background rate of chromosomal lesions are leading to this. No, that does not make sense. I guess you would have to have really huge number of cell divisions to generate that kind of the kind of lesions we see. We also suddenly see large, what we call macromutations or hopeful monsters show up in Barrett's esophagus suddenly. But I don't have a good quantitative argument. Just a argument by intimidation. All right. This is a figure from Noemi's expands paper, which is just to say that there are much more sophisticated ways of estimating the within sample diversity, and you should go see her poster, which is on the first board out there, where she's applied this to the TCGA data. Okay, so that's shown you a little bit about measuring mutation rate. I've shown you a little bit about measuring, actually quite a bit about measuring diversity or intratumor heterogeneity across tumors, and that's the one we're most excited about, trying to generalize across cancers to show that it's a universal biomarker. We're also very much interested in measuring now how diverse the microenvironments are, and there's good ecological theory that says that diverse microenvironments should select for metastasis as well as genetic diversity. And so here's some of the theory. So we did some computational modeling where we were able to control in the model the degree of microenvironmental heterogeneity, or actually the degree of patchiness of resources. And the intuition here is that if resources like glucose or oxygen are patchy, that is, some areas of the tumor have very low and some areas of the tumor are very high, then you get selection for cells to migrate to find the rich areas of resource, right? Whereas if all the resources are uniform, there's no selection on cells to migrate. They can't do any better by moving. And so if we allow migration rate to evolve in a simulation, and we can tune up and down the amount of patchiness, both temporal and spatial heterogeneity resources, what you can see on the right is the evolution of the migration in one of the runs of the model, that the migration rate goes way up when you have temporal and spatial heterogeneity resources. Athena Actibus did a related model here, which is to show that even if the resources are being delivered homogeneously across a tumor, if the neoplastic cells are overexploiting their environment, that is, if they're taking up more resources than are being delivered to any part of the tumor, they will locally degrade and deplete the resources, and they'll cause this patchiness of resources. So if the neoplastic cell has a ramped up metabolism, it will be under selective pressure to be able to move because it uses up its resources, and if it sticks around, it dies. Right? So she simulated this. She simulated the parameter was the relative metabolic rate. So compared to a normal cell, how much more resources is a neoplastic cell using, and showed that when you tune that up and watch the evolution of migration, that when cells that are requiring more resources will evolve to move more likely. This all comes out of dispersal theory from ecology, which says that if there's patchiness resources, you get evolution for dispersal. And we think this really is the right framework for understanding the evolution of metastasis. Once you can measure these universal biomarkers, then you can actually start talking about targeting them. So we're very much interested in doing cancer prevention and also delaying relapse by trying to slow the process of evolution. So that leads you directly to thinking about, well, can I reduce the mutation rate? Can I reduce the genetic instability? Can I reduce the stem cell population size? This is our traditional approach, right? We try to either surgically resect to reduce population size or use chemo to try to kill cells to reduce population size. But we should also be thinking about, can we slow the generation times to make the generation times of the stem cells longer? That would also reduce the evolvability of these tumors. And can we slow clonal expansions? These are essentially our questions that haven't really been addressed yet. Similarly, can we prevent metastasis by homogenizing the microenvironment, that is, preventing this patchiness to reduce the selection pressure for cells to move? And the figures in the lower right are not my work again, but it's a dramatic example. These mice that were PhD2 hemizygous mice actually normalized the oxygen in their tumors. You have very smooth distribution, homogeneous distribution of oxygen in the tumors, and sure enough, the lymph node metastases and the lung metastases went way down.
So is there a kind of implicit assumption in all this that the stem cell populations are a genotypic state, not a phenotypic state?
So the question is there an implicit assumption that the stem cell populations are genotypic rather than a phenotypic state? No. And so, yeah, so the stem cell world is a mess from my perspective. It's very hard to study. It's all operationalized based on xenograft models. There is evidence that in some cases that the non-stem cells can become stem cells, and if that's frequent enough, then effectively all the cells in the population are contributing to the future of the population, and so I would consider them all effectively stem cells.
Can I ask a question in a way that avoids the stem cell?
Okay, sure.
So one can ask more generally if one looks at where you see substantial phenotypic diversity in the tumor. And since you see homogenetic diversity as well, the obvious thing, okay, that's because of the genetic diversity is why it's phenotypic diverse. But it can presumably also be phenotypic diverse primarily for developmental reasons, like our tissues are phenotypically diverse. And what is known or ways of studying the differences between those two, the balance between those two events?
Okay, so if I can summarize and probably destroy your question, the observation is there's clear genotypic diversity within tumors and clear phenotypic diversity in tumors, and what is known about the relationship between those, because they're not necessarily the same thing.
Particularly if the phenotypic is more like developmental.
Particularly if the phenotypic diversity is, yeah.
You mentioned the
Right. Right. So this clearly is a fair amount of phenotypic diversity that is not genetic, that is differentiation programs that are being activated in one way or another. And what is known about that? Well, unfortunately, that doesn't avoid the stem cell world, because that's, I think, in large part what it's all about. So I'm going to have to punt. I don't know. I think in large part there's certainly open questions there. Let me just say that. Okay. So besides, so it looks like there's evidence that if you could homogenize the microenvironments, you could reduce the metastasis. Or presumably based on Athena's work, if you could prevent overexploitation, and by the neoplasm, you could also prevent metastasis. And in general, we should be thinking about reducing the selective pressures on cell migration. So the sort of final question I want to get to is if intratumor heterogeneity is such a problem for us, there's also an opportunity here. It means these tumors are very evolvable, and so we should be asking, how do we want them to evolve? What selective pressures, what do we want to select for? Right, I think that's one of the major questions we should be thinking about in terms of therapy and control of these tumors. So how can we exploit intratumor heterogeneity? So Bob Gatenby has this fascinating result called adaptive therapy, and the sort of figures in the upper right-hand corner. The idea is not to try to cure tumors, but try to keep them a stable size. So if they grow, prune their branches, keep them to a stable size. The figure from the lower corner is from their adaptive therapy paper, showing that if you treat the tumors to keep them a stable size, raise the dose if they're growing, lower the dose if they're not growing. That's the lower green line there. They were able to keep the mice alive indefinitely with these very aggressive xenograft of the AVCAR3 cells. They repeated this experiment, and now they've done it in a triple-negative breast model as well. It hasn't been published yet, but it's working there as well. So that algorithm is actually based on trying to play off chemosensitive cells with chemoresistant cells. And the assumption it's based on is that resistance has some cost. So in the absence of a drug, the sensitive cells should outcompete the resistant cells. And in the presence of the drug, the resistant cells should outcompete the sensitive cells. Gatenby also has the idea of double bind therapy, which is if you could apply two different drugs or two different selective pressures that select for opposite phenotypes, then you would put the tumor in a double bind. That is, selection for resistance in one drug makes them super sensitive to the other drug. One of the ideas we came up with is related, called a sucker's gambit. That is, if you could sucker the tumor into a vulnerable state by selecting against resistance initially, that is benefiting the sensitive cells, drive the resistant cells extinct, then you have a very vulnerable tumor and you can come in with your drug and you no longer have resistant cells there. The related approach we also, in the same paper, we looked at was called benign cell boosters. So if you ask the question, what do you want to select for, we would like to select for a benign tumor. So can you increase the fitness of benign cells relative to the more aggressive cells? And that paper is all simulation, so it's not yet anywhere near the clinic. And I want to show you some final unpublished data, which is another approach to exploiting intratumor heterogeneity. And it was actually mentioned yesterday early on, this idea of selective interference. That is, different clones that both have a selective advantage will come into competition with the population and will inhibit each other. And can we exploit that? So the observation here is that if you've got a single pressure on a population, and like in the upper panel here, that's selecting for the red cells, then the population will adapt pretty quickly, and mutations responding to the red pressure will expand and take over the population. However, if you apply multiple selective pressures, then mutations to the different pressures can actually interfere with each other. And so even though you have mutations to the red, the yellow, and the blue pressure showing up in the second petri dish here, only one of them eventually is going to win out, and it'll be as if there was no evolution that happened with respect to the red or the yellow pressure. Right? So that's the theory. It was actually developed. The theory was developed by Nick Barton in the context of what are the limits of natural selection. But we would like to limit natural selection. That's a good thing for us in cancer. It's particularly effective in asexual populations that can't recombine these different mutations, adaptive mutations. So that's also good for us in cancer, because as far as we know, it's reproducing asexually. But no one has really directly tested this theory. It's been used to explain a lot of results, but not directly tested. So we did that. We grew yeast for 500 generations. These were asexual haploid yeast. We had 3 different selective pressures. It was the carbon source, glucose, an amino acid. They can't produce their own histidine, so we could titrate the amount of pressure on the population by how much histidine was in the median. And also, they couldn't produce their own uracil, so we could titrate this nucleotide source as well in the medium. So we can evolve the populations of yeast under either one or all three pressures, and then test the fitness at the end of this process with respect to one pressure to see if the population under three pressures was slowed down in its evolution toward that pressure. So here's sort of the experimental design. It's pretty straightforward. You take an initial culture, you split it into these different populations. We had replicates for each of these, either a single pressure, the top three, or all three pressures, the bottom row here, which we call the all minus. So we got the glue minus, the his minus, euro minus, and the all minus. So here's some of the fitness data. At the end of the process, each data point is an independent measurement of fitness. And you can see that if you measure fitness under just limiting uracil, then the red box here shows that those populations that were only evolved under uracil restriction had a higher fitness than any of the other populations. And in particular, we're comparing it to the all A and all B on the left-hand side. Those are the cases where the populations were exposed to all three pressures, including the uracil pressure, but clearly didn't evolve as quickly to the uracil selective pressure. Here's a summary of all of the data. So on the diagonal, the nice red area is showing that the fitness level of populations that were exposed to only the single pressure that they're being now measured on had the highest fitness. Okay, that's what we expect from evolution. The interesting bit here is, let's see if I can do this. Yeah, these rows here were all A and all B. Those were the populations exposed to all three pressures. And if you measure them against one pressure, you can see they have not evolved as much as the populations that only were selected under a single pressure, i.e., we have been able to slow down evolution by applying independent selective pressures to the population.
How did you measure fitness?
How did we measure fitness? We had a reference population that was antibiotic resistant or anti-yeast resistant, antifungal resistant. And so we could measure, we could put them in competition over 24 hours and then measure how much the evolved population expanded relative to this reference strain. They're just parallel. We just repeated the experiment in parallel, so they're just two different independent runs of the system. Yeah, so all B is sort of, it's a little bit weird. It did, well, sorry, let's see. It did, I guess all A did pretty well in uracil, but yeah, which mutations show up and how the population evolves, of course, is stochastic, and so you don't get the same answer every time. Right, so in conclusion, this intertumor heterogeneity is the bane of all of our attempts to characterize tumors. Right? It's a real problem for us, both scientifically and clinically, but it's also the key to understanding how these neoplasms are changing and responding to our therapy. And measurement of that intertumor heterogeneity appears to be a robust biomarker. So far, evidence is that it doesn't matter which metric you use, and it doesn't matter which assay you use to characterize clones. Yet we don't really know the dynamics of this heterogeneity or how it changes over time or what drives that change. But we think that the measures of somatic evolution in general and ecology are universal biomarkers and can be targets for intervention, particularly for cancer prevention and for slowing relapse. So let me just thank my wonderful collaborators and support, and also mention that we'll be hiring a bunch of postdocs and staff scientists in the near future doing these different projects, and I hope if you're interested, you'll contact me. Thank you.
Our experiment worked. The questions during the talk were really great. So open it up for more discussion.
Yeah, can you use a microphone? I think that'll save me from butchering your question by repeating it.
Hi, Joel Neal from Stanford.
Okay.
So your yeast model has interesting implications for the treatment of patients. And one concept for treating patients is use a bunch of things all together, which ends up being less tolerable for the patient and maybe intolerable for the tumor too, hopefully. The other strategy that we've used more often is rotating amongst different therapies. So it seems like another experiment might be to then take the individual selective pressures and see if the yeast are equally unfit at the end of seeing all three in sequence as opposed to all together.
Yeah, this is a really important question, clearly, in therapy, whether to apply drugs in parallel or in series. And the evolutionary biologists in the audience will probably cringe at the idea of doing it in serial because it's sort of easy to evolve in response to with one pressure. But it's a cost-benefit analysis of the toxicities of putting the drugs together, as you well know, and whether you have to reduce the drug levels. So I think in general, it's going to have that question itself of the serial versus parallel has to be done for any set of drugs experimentally, although I think you can do some good modeling. And one of the experimental models here would be to do it in yeast, and we haven't done that. It's an interesting question and an important one. I should mention that if you take the implications of this experiment, you don't have to use a toxic pressure, right? You can delay evolution by putting a nutrient in that's new or special, that you can get specialized. You can put in anything that's totally non-toxic as long as it's putting a selective pressure on the population. It doesn't have to be killing. It can be helping the more benign ones or whatever. So it could be a high broccoli diet could be one of your pressures. We don't know. But this is one of the things that's exciting to me is that we're not necessarily talking about combining toxic pressures. But it's a great question and an open one, unfortunately. Yeah.
Yeah, we get asked all the time in clinic about reducing glucose and alkalinizing the diet and the high broccoli and things like that.
Right.
It sounds like the bottom line is if you're going to do it, do all of them.
Yeah, right. Yeah, particularly the non-toxic ones. Yeah, I would agree. Yeah.
Presumably with high broccoli diet, one has to worry about quality of life issues as well.
Indeed.
So
maybe a high chocolate diet?
Yeah, that's okay. So you brought up some very good points at the beginning about the importance of the sort of big populations and what goes on there generally. And then also at the end about the potential analogs or usefulness of trying to understand some of the microbes. So I sort of wanted to add to that and also connect maybe back to Ken Kinsler's talk yesterday. And that's a question about necessity and sufficiency of mutational changes. If there are some particular mutations, even a particular point mutation that is really going to be sufficient by itself, in addition to perhaps being necessary, then that's going to happen so fast that things will take off and one will get the cancers the whole time, or they'll rapidly develop further once they start getting somewhat uncontrolled growth. And so that sort of very strongly suggests that a lot of the things which maybe are all the background mutations which are not common to a lot of the within the tumor, that they're much more diverse, that they may be something else, and some fraction of those, maybe only a small fraction, may be important. And so for this, it seems there's some analogy which is perhaps useful with looking at the evolution of HIV within individual hosts. So the sort of classic view, right, is that there are important mutations that are ones which beat a drug or that change how the immune system sees it, and that's mainly what goes on, and then everything else is just sort of neutral drift and just random mutations. But this is one where it really now appears that that's not correct, that what seems to be going on is that the population is evolving the whole time. The host doesn't care about that evolution, but the viruses care about it a lot. They're competing with each other and so on. Eventually, they probably become diverse enough, the population, that some of those individual viruses have some of the enabling mutations such that the negative side effects for them of the resistance of a drug resistance mutation or immune system beating mutation are sufficiently low that they can take off. So there can still be, again, individual mutations that in some sense are the ones that convey the drug resistance or convey the becoming a cancer here. But the other ones can still be very important, and the diversity helps enable those just by random chance getting some of the mutations which will compensate later on, also enable in a broad sense for the really key ones.
Right.
And that is a very general thing that happens in big populations.
Yeah, no, I agree.
Carlo,
Yeah.
absolutely wonderful talk.
Thanks.
I really enjoyed it. I wanted to help you out with one thing. You mentioned that you were about the instability of chromosomal instability.
Yeah.
And there is some decent experimental data for that. In the case of colorectal cancer, if you assume the rate of chromosomal loss in tumors that have, you assume Occam's razor, or there's only one type of instability going on. If you compare microsatellite unstable tumors to the vast majority of tumors which are microsatellite stable, they have about 100-fold increased rate of chromosome losses. Now you could make some arguments.
By rate, do you mean incidence when you measure the amount?
Actually a rate.
Rate, okay, per
By
cell
rate,
division.
you can actually measure rate using cells in culture, comparing
Okay.
to cells in culture that have a mismatch repair defect and have a very high rate of small mutation rates. If you
Right.
compare those cell lines that do not have that instability, they lose chromosomes at about 100-fold higher rate.
Right.
And of course, that is also manifested as aneuploidy,
Right.
whereas the mismatch repair tumors tend to be stable.
Right.
One could argue that perhaps that the high mutation load of the mismatch repair is selecting against loss events, but on the other hand, these rates of loss that one sees are consistent with what one measures using markers in animals.
Mm-hmm. Great.
The second thing I'd like to say is I like your point about a genome is absolutely technically correct and absolutely true, and it reflects the problems of trying to characterize these tumors. On the other hand, there is also a shared genome.
Yeah.
That all, since these are derived from a clone,
mutations,
that
right.
they do share some critical events,
Yeah.
and and if you look at metastases, in some, I know there's exceptions, but if you look at metastases in colorectal tumors, some of the key players are shared amongst those tumors. So that's the other half,
That's
the
right.
half full cup half full view.
Yeah, right. I agree. In fact, if you think of this phylogenetically, what you see from these assays are the deep bits of the trunk usually, or the early branches, because those are the ones that are shared by enough cells that you can pick them up.
Yeah.
And so it is a way, and that's valuable information, of course. What were the things that were in that cell that initially did the transformation? So I agree. Maybe I was a little unfair about it.
Yeah. And your point, your excellent point you made about the evolutionary fuel in a tumor being greater than that of Homo sapiens is absolutely true. But we can look around the room and identify us as humans. We can identify the birds, the bees, and so forth. So there are some commonalities.
Sure.
Yep.
Yeah, no, that's important. And I guess we talked about this a lot yesterday, the importance of understanding the regularities and the constraints on this system. Yeah, I completely agree. Charlie?
I think David's first.
Oh, sorry, David.
Pushed in.
Yeah, there's a theory in ecology where if ecologists look at island archipelagos, the number of species is proportional to the size of the island.
Right, as island biogeography.
Yeah, does this hold up if you look at the archipelago of a metastatic tumor? And does it hold up from patient? Does one patient's archipelago have the same relationship as the other patients?
It's a great question, yes. Or there does seem to be this nice correlation between, or actually function that describes the number of species versus the size of islands, and it just hasn't been measured. You could do this exactly the same thing if you had really good measurements of the clonal diversity of mets and the different sizes in the primary, but it hasn't been measured yet.
And perhaps you could use it as a tool to know whether your biopsy has really captured, a high percentage of the...
Yeah, right. You'd have to be careful about how you actually biopsy. You want to sort of grind up the whole thing in that case. But yeah.
It was a cracking talk, Carlo. Thank you. I just wanted to sort of add a point
I
about
appreciate the Wallace and Gromit reference there.
Yeah, so it's an English phrase. Apologies. Your poster outside alludes to more complexity about the relationships between heterogeneity and outcome.
Yeah.
Do you want to comment on that? How that fits in with your indices, and whether there might be some problems implementing this biomarker in a clinical setting?
Right. So one of the things that Noemi did was looking at TCA data was actually try to ask if there's any relationship between the number of subclones that we're detecting and indications of the amount of microenvironmental diversity. Now, we don't have great measures of microenvironmental diversity in the TCA data. What we have is the frequency of different kinds of lymphocytes and stroma and whether there's necrosis and stuff. So we tried to put some measurements on those environmental measures, and there is some relationship. Noemi, do you remember what the correlation is there?
Across all tumors, it was about 4.22.
So there's about 0.22 correlation across all tumors. This is 12 different cancer types that we were collecting together 1,000 different tumors.
Yeah.
So there does seem to be some relation, even by these very crude measures. But more interesting would be to actually get some spatial, so in situ measures of microenvironment.
And then the issue about the risk of relapse correlating with sort of an intermediate threshold of diversity rather than the most extreme levels of diversity.
Yeah, I didn't touch on this. So one of the things that Charlie has done and Noemi has followed up on is that there's some indication that the most diverse tumors actually have a better prognosis. Or I should say the tumors with the highest mutational burden or instability burden have a better prognosis than the moderate levels of burden. And the low levels of burden also have a better prognosis. So the extremes can be better than the middle for these tumors. So Noemi found evidence of that across all these different cancers as well. Both the burden, the amount of copy number change, and also some estimates of the diversity.
Do you think that's immune-mediated, or do you think that's autonomous lethality, the mutational meltdown?
Right. So the question is, why are these really high-burden tumors sort of better prognosis? So one is possible that when we apply a mutagen, like a PARP inhibitor or just a cytotoxic, that we're sort of pushing these tumors over the edge of the error catastrophe threshold. That is, they're already, so many of the cell divisions are broken because they're producing so many alterations that if you just add a few more alterations, then not enough divisions are viable to actually maintain the tumor. So that's the meltdown argument. Another one is that there's the immune system, that these really diverse tumors or really highly mutated tumors should be having a higher immune response. We don't know that, right? So it's a great open question.
But the time courses on that help the next section.
Right.
The time courses should help.
If you could watch that process over time, yeah, right.
Two questions. Excellent presentation. First question, the microenvironment, very important. Do we have recommendations about not only taking tumor biopsies, but biopsying the surrounding normal tissue, and then what should we quantitate?
Yeah, so those are all open questions. So my understanding of this is that we are getting the microenvironment in the kind of samples we're getting at the moment, that we're seeing.
Unless you're doing single cells.
Yeah, right, unless you're doing single cells. So the kind of in situ stuff that Nelly was talking about yesterday, I think is really valuable. And yeah, I guess from the tissue blocks, you should be able to see a lot of the microenvironment. But if you only took a sample from the center of the tumor, that you might be missing critical stuff that's happening at the periphery. So that's one issue. Then what should we be measuring? That's a totally open question, but dispersal theory argues that it's the heterogeneity of the resources, so we should be looking at hypoxia and maybe glucose and whatever else.
Vascular density,
possibly,
or
yeah.
Immune infiltrate, and which immune infiltrate.
Yeah, so for me, one of the open questions is to be able to detect when is the immune infiltrate acting as a predator and causing external mortality versus when is it being co-opted to send survival signals and things like that. And as I understand, it's actually pretty hard to detect whether there's actual predation happening. That's one of the things I would like to be able to study better, particularly in the pre-malignant tumors. I think it's relevant, but.
And the second question is a little more controversial, but it has the chicken and the egg controversy. Does a bad initial tumor with whatever you want to call driver mutation develop more heterogeneity, and that's just a marker of a bad tumor? Or is the heterogeneity of itself bad? And maybe we can track back the most heterogeneous tumors and see if we can identify in the early truncal changes whether it's the chicken that's bad right from the get-go and not something that happens later on when you have all the different pressures from treatment, et cetera.
All right, so I think on at least theoretical grounds, you'd have to say that the heterogeneity is not just an irrelevant side effect, that it is driving a lot of this process.
Well, I'm not saying it's irrelevant, I'm just saying it's determined by your initial mutations.
So no, it's got to be the case that some mutations can cause, we know this can cause a mutator phenotype, and so prior to that, you can imagine there's got to be many tumors that don't start with a mutation and that happens later. We certainly see that in Barrett's esophagus. Now, one of the interesting results that's both in Barrett's and looks like maybe happening in AML also is that we don't see a sort of a monotonic increase in diversity over time. And when we take 2 time points or more in Barrett's esophagus, it looks like it's pretty much stable, and if anything, maybe going down a little bit over time. When I did these initial studies looking at diversity predicting progression, I thought, well, this is predicting progression because it's going up over time, so when we see a high diversity, it's because they're late in the process and they're almost at cancer. But it actually looks like that's not the case. It's that some of our patients are maintaining a high diversity. They have maybe a high mutation rate, and some of our patients have a low diversity, and that maintains at a low diversity. And so there's some somewhat stable turnover in the diversity, but relative to the absolute levels looks like it may be stable. Now, these are just hints so far. We don't have this in many tumors. We're not even sure of it in the tumors we got it on, but that's how the picture is shaping up, I think.
Thank you.
Yeah.
Thank you for your talk. My question was related to actually, most of the times I've seen diagrams of clonal populations competing with each other, but do you have any evidence for, or how possible is it to look for example, cooperativity between the clones over time? So are there relationships amongst the subpopulations that are preserved over time? And do you see any of that, and how can you?
It's a great question, and the answer is we don't have a good way of doing that yet. So Ken Pienta and the Axelrod brothers published one of the original papers in suggesting that cooperation between clones could be a really critical aspect of cancer that we haven't really looked at yet. So one of the things I thought was most interesting about Nelly Polyak's talk yesterday was showing that minor population that was producing IL-10 was acting as a IL-11, sorry, IL-11. Yes, sorry, my apologies, IL-11. Was showing this kind of cooperation along with the free rider effect. The other cells were getting the benefit of it without having to produce the IL-11. And so the beneficial mutation was not able to spread to take over the tumor. So that's one aspect of cooperation that exposes you to free riders and cheaters. But I think you're asking about mutualism, right? Where
yes,
both
actually.
clones are benefiting each other.
Yes.
So theoretically, it's quite possible, and I'd argue that it's likely to happen, that you could have a growth factor producing clone and it's another survival factor producing clone, or an invasion, like metalloprotease producing clone that opens the door for everybody else. And there's all kinds of ways that one clone could be benefiting another. And you can potentially get this mutualism, but it basically hasn't been studied yet. I mean, there's a few labs. Ken Pienta's lab is trying to do it. Nelly is obviously doing it, but we just don't know yet.
Okay, thank you.
Thanks.
I was struck by the robustness of the observation that genetic diversity as a biomarker for kind of outcome here. In the context of, we know that for a given set of tumors, if you keep the tumor stable, the tumor type kind of fixed, we know that not all mutations are equivalent in that context. And so how do you reconcile those two things, of the fact that you see this robust effect of genetic diversity across even different tumor types, yet knowing, are those events so rare, or the ones that have those marked effects so rare that they're swamped out by a kind of overall effect? Or how do you reconcile those two things?
No, I think there's maybe two aspects to your question. One is why can you look at diversity and not just the driver mutations? Is that saying that?
We know that, so say you get, particularly for low numbers of mutations, we know that different drivers, particularly on the two phenotypic outcomes that probably drive a lot of what you're seeing of division rate and mutation rate. I mean, P53 probably being one of the best examples, and any number of well-characterized oncogenes. We know that there's a dramatic effect in tumors with mutations in those versus without, particularly when the numbers of mutations are small. Yet you're seeing a global effect of just overall number that isn't yet explained by.
Okay. Yeah. Let me be clear that I'm certainly not suggesting that all mutations are equal. That clearly some mutations have a big beneficial effect for the clone. Some are neutral. Some are deleterious. Why is diversity a robust or good predictor? I think that diversity highly correlates with the likelihood of getting those important driver hits that are increasing the selective advantage of the clones. That's my intuition, but that's not why it is.
And I guess the correlate of that is, so if, kind of apropos of the questions asked earlier, is that are you actually uncovering a more subtle, not a traditional mutator phenotype of a clearly identified gene or gene target that regulates DNA repair or cell division, but are you uncovering kind of more of a global effect of mutation rate? Is it really the genetic diversity per se, or is it what's leading to the genetic diversity that's underlying the difference in behavior?
I think probably the latter. In fact, one of the things I think is a benefit of genetic diversity is it integrates both the intrinsic causes of diversity, like the P53 hit, and the extrinsic cause of diversity, like tobacco exposure or UV light. And so you get to integrate all of these causes that are driving the process in one readout. That's my intuition.
And I guess the last thing I'd say is that, so the part that's also intriguing is that our treatment, particularly with traditional chemotherapy, is also superimposing, with traditional chemotherapy that causes DNA damage, is superimposing sort of an interesting facilitation if there's an underlying kind of subtle mutator phenotype, but it also might explain why at the high end you were just mentioning that, you know, at both very low and very high levels of mutation that those folks do better outcomes. I mean, some of those very high rates can be seen with familial cancer syndromes. And in those folks, they actually respond quite well to traditional chemotherapy because those cells are quite sensitized. So can you comment a little bit on how the treatment part superimposes on top of the genetic?
Yeah, I mean, I agree. I have the same intuition I think you do, that these high mutator load tumors have a hard time accepting all the new mutations that we're imposing from our chemotherapies. It also is really scary to me when we were looking at the pre-therapy and then the relapse tumors. We see so many more. It looks shaping up to look like there's so many more mutations in the relapse that it looks like our therapies are probably generating a lot of the evolution that drives the relapse, and that is really scary to me.
So, would you like to comment on, I think, a couple of the questions today and a couple of the talks yesterday on the concept of cryptic genetic variation, where a mutation occurs and the background mutations are neutral, but then when that first mutation occurs, some of those background mutations now have a phenotype? And
yeah.
Is that playing a role here?
Sure. So I mean, the easiest thing to think about and see is that there's probably that a resistance mutation may well be completely neutral until you bring the drug in, right? And then it suddenly reveals this, what was otherwise cryptic evolution. You're actually referring to more of the stuff like with HSP90, where there's this buffering of chaperone molecules that fold the proteins in the correct fold, even though the background genetics is quite different. And then when you suppress HSP90, you get all these interesting mutations. Yeah, so I'm interested in that, and I think it's an important thing to explore. And my intuition, of course, is that when you do suppress those buffers, that you will reveal a lot more heterogeneity, and then you actually increase the rate of evolution. And so what we should be thinking about is increasing the buffering to try to keep the phenotype in a more normal state, even though the genetics underneath are getting mutated.
But maybe an HSP90 inhibitor will push you to that extreme.
Yeah, maybe. Yeah, it's an open question. Maybe if you do HSP90 and mutagen, and you can push them over the cliff of their catastrophe threshold. Yeah, it's possible. All right.
Discussion.
Thank you.